Hi everyone, I am Dr. Anisha, your microbiology faculty, and in today's session we will be covering up the lab diagnosis of tuberculosis. And under lab diagnosis, we will be talking about different specimens that are required, their microscopy, different culture methods, automated methods, and certain molecular tools and techniques which will help in diagnosis very early in the infectious stage. So to begin with, we first need to talk about the diagnosis of active tuberculosis cases, whether it is pulmonary or extrapulmonary cases. So the very first thing that we do for the diagnosis is the specimen collection. And what kind of specimens do we require? First and the foremost, and the most common one, it is the sputum sample. And what kind of sputum is mandatory or it is a prerequisite for the starting of the diagnostic procedure? It is the mucopurulent kind of sputum. So what do? We ask the patient to inspire deeply and we ask him to cough out deep during exhalation, and he has to collect that sample in the wide-mouthed screw-capped container. So deep inspiration, it is very, very essential. So that is the reason why we are asking the patient, because we require mucopurulent sample. Most of the patient at times they send over or they collect salivary samples. So we are not going to reject, we are not going to accept those samples, whereas we are going to reject salivary samples. So mucopurulent nature, it is the prerequisite. And how many samples do we require? So we require 2 samples. One is the spot sample and the other sample, it is the early morning sample. And alternatively, if in case the patient is unable to provide the next day early morning sample, so we will collect, alternatively, we will ask the patient to collect the sample. So we'll be asking him to give us 2 spot samples at least 1 hour apart. So that is how we are going to ask the patient to give us sputum sample. Then comes the gastric aspirate. And in what conditions are we going to take the gastric aspirate sample? So it is in case of children who have this tendency to swallow the sputum. So in this case, we'll be taking up the gastric aspirate and we'll be using for the diagnosis of TB, pulmonary TB. And the third case, it is the extrapulmonary TB samples. And these samples will be collected based upon the site. Right? So these are the most common samples, most common collections that we can use for diagnosis. Then the next step that we do, it is the digestion, decontamination, as well as the concentration. So why do we require digestion? Digestion, it is for liquefaction of the thick mucopurulent sample that we have asked the patient to give us. It is for liquefaction of sputum. The next one is decontamination, because we want to get rid of the normal flora. We do not want any contamination in that particular sample, right? And then the concentration. Concentration, naturally it is in order to increase the yield of the sample. And this particular step of digestion, decontamination and concentration, it is not required in cases of automated methods. Not required for automation. So what are the two methods that we most commonly use for this particular step? So these methods are number A, modified 𝗕𝗏𝗏's method, modified 𝗕𝗏𝗏's method, where we are using around 4% NaOH. So what do? We first take around 3 to 5 ml of sputum sample and we mix that in equal quantities of 4% NaOH. And we will leave it for around 30 minutes in order to liquefy the sample and to kill the normal flora. And then what are we going to do? We are going to centrifuge it and neutralize it with distilled water. And again centrifuge it. So finally we are going to have supernatant and the deposit and the sediment. So the supernatant would be discarded off and the sediment would be used for culture. So this method, it is more favorable for conventional culture methods. Then we have the second method wherein we are using N-acetyl-L-cysteine and NaOH, 2% NaOH, and this method, it is favorable for automation. And what are the two steps that we are doing? So N-acetyl-L-cysteine, that is going to liquefy the sputum. And NaOH, this is going to kill the bacilli, kill the normal flora, bacilli. So these are the two methods which are used for the digestion, decontamination, etc. Then coming to the microscopy of the sample. So for microscopy, we will be requiring staining methods, which are hot method, cold method, certain other modifications, etc. Right? So what is the hot method name? It is the Zn staining, the conventional Zn staining. And the second method, it is the cold method, which is known as the Quinones method. Right? So for staining, the important steps of staining, and to remember these steps for Zn staining, we have a mnemonic which is cough has sputum. So I have highlighted these letters for you to learn the different steps. So with C we have 𝗘𝗅𝗏𝗏 as primary stain. Then with H, we are denoting the intermittent heating. What kind of heating? Intermittent heating. With S, we have the decolorizer, which is sulfuric acid or H2SO4. This is the decolorizer. And finally with M, we have the counterstain or methylene blue. Which is the counterstain. So whenever asked about the Zn staining, we know it is done for diagnosis of TB, and for TB we require sputum sample. So this is the easiest mnemonic that I could get for Zn staining steps. Cough has sputum. Now then comes the cold method, which is the Quignon method. So how do you remember Quignon and what are the different modifications which are done for the cold method than the hot method? First things first, so Kim Jong-un, he is the dictator in North Korea. Right? First thing. Then we are highlighting North Korea and it is pretty cold. Pretty cold in Korea. So from here we derive the cold method, first thing. Then un. So this particular method, it is unheated method. We do not heat the sample. This is one modification. Then this word jong, does it sound similar to long? Long. So we have long staining time. Staining time with the stains, with the different stains. And what stain? It is the 𝗘𝗵𝗆 duration. It is the 𝗘𝗵𝗆 duration, which is longer. Long staining time or long staining duration, it is with 𝗘𝗵𝗆. This is the second modification. And the third modification is that now this guy, Kim Jong-un, most of you might have seen him over television, that he is kind of quite bulky. And in Hindi, we use this word goal, no fat shaming from my end, but it is just for the mnemonic. So, goal will be used as a mnemonic for a component phenol. Goal and phenol. So, phenol concentration in 𝗘𝗵𝗆 is increased. So, this is the third modification. First is unheated method. Then there is long staining time. Second thing. And third, where phenol concentration, it is increased in 𝗘𝗵𝗆. So these are the three important modifications done for cold method. Now let's talk about the other modifications which we use for the staining or different diagnosis or different purposes. So what are the modifications? Firstly, acid alcohol. Acid alcohol, which includes 3% HCl and 97% of ethanol, ethyl alcohol. So this modification is used to differentiate Mycobacterium tuberculosis from Mycobacterium smegmatis. And how is it differentiating? So Mycobacterium tuberculosis, it is acid plus alcohol fast. Whereas Mycobacterium smegmatis, it is just acid fast but not alcohol fast. So smegmatis, it is majorly found in the renal TB. So this is the first modification we can use. Also, one important thing that I forgot to mention about the ZN staining are the time durations. Firstly, 𝗙𝗵𝗆 primary stain. Primary stain, it is done for 5 to 8 minutes. Then decolorization, 2 to 4 minutes. And the counter stain or the secondary stain, it is for 30 seconds. These are the different durations. And also, what is the concentration of H2SO4? So NTEP says it is 25% concentration of H2SO4 which is used for ZN staining method. One thing. Now let's talk about the other modifications. Other modifications would include the change in concentrations of H2SO4. So while changing the concentration of H2SO4, a very recent question gets asked, which is modified ZN stain. And in this particular method, what is the modification? It is lowering the concentration of H2SO4 from 0.5 to 1% for staining of different other species or different bacteria. What are the different species? So for 1% it is used for nocardia staining. 0.25 to 0.5% it is used for spores as well as Cryptosporidium. Right? And another modification which is done, it is the use of 5% H2SO4 for staining of Mycobacterium leprae. So these are the different concentrations, different changes in concentrations that are used for different staining purposes. Now let us talk about other modifications which have been done. So firstly, we spoke about the conventional classic ZN staining. The other one we had was Quignon method, which was a cold staining method. And under cold staining, we have another method which is the Gabet's method, wherein the decolorizer and the counter staining is done in this same step itself. Decolorizer plus counter staining is done in one step itself, right? And then we have spore staining method. Spore staining, which is done by Muller's method. And 𝗕𝗵𝗄𝗏 method. Then for tissue sections, we have 𝗙𝗵𝗿𝗺𝗏 stain and 𝗕𝗵𝗿𝗺 method. So these are other important modifications which can also be asked in your exams. So now moving on to another very important staining technique which is recommended by NEEP also. It is the fluorescent staining method. So what are we doing in this particular method? We use 𝗤𝗏𝗏 solution as the primary stain. And it is done for around 7 to 8 minutes. Then we have acid alcohol, which is done for the decolorization is done for 2 minutes twice. The second step, it is the decolorizer step. And then the third step is use of potassium permanganate. For 30 seconds, right? And after that, we will be observing the slide under fluorescent microscope. And what do we observe under fluorescent microscope? So the bacilli, they appear brilliant yellow in color. Yellow, and they fluoresce. They show fluorescence. So one major disadvantage over this particular method is that certain artifacts, there can be a confounding effect because of certain artifacts which can also glow under fluorescent microscope. So this is one disadvantage. And the other disadvantage of the staining method, that is fluorescent staining and Zn staining, is that they do not guarantee viability of the bacilli. Viability of the bacilli. But one major advantage associated with fluorescent staining is that so the turnaround time is going to be less. So because of the fluorescence, you are able to detect better and you're able to screen large number of fields in a considerably less number of time. So this is less time consuming. But for this also we require an expert to look into these slides. Right? So this was about the fluorescent staining, which is recommended by NTP. And then coming to the next step, which is the culture. So what are the conventional culture methods? most of you by now know that what is the most common culture media that we use? It is the Lowenstein-Jensen media. LJ media, which has malachite green as the selective agent. This is one media, and this is the most commonly used and recommended by NTP. What are the other medias? We can also have Dorset egg media, and then certain other agar-based medias can also be used, but they are not recommended. These are Middlebrook 10 and 11. So LJ media, Dorset egg media, these are all agar-based medias. And what kind of colonies do we get on LJ media? These are rough, the most famous question from the culture of TB, rough, tough, and buff. What do we understand by rough, tough, and buff? These are rough in texture, these are tough to emulsify and break, and these are buff colored. Buff colored. So this is the typical picture for the culture or the colonies of Mycobacterium tuberculosis. Tuberculosis, rough, tough, buff. And Mycobacterium bovis, they have smooth, white and creamy colonies, and they easily break off. Right? So for automation, what is the most recommended and what culture media do we use for automation? It is the Middlebrook 9, which is the most common and most recommended and mostly used in automated culture medias. So it is liquid broth. Liquid broth. Right? So let us now discuss about the benefits of automated culture methods or automated liquid culture. So the very first benefit is that they monitor the growth continuously. And they have a very short turnaround time. They have a shorter turnaround time, which is around 3 to 4 weeks. So if in case there is any growth, it gets detected within 3 to 4 weeks, which is not the case with the conventional culture medias like the agar-based medias. When we use suppose LJ media, it takes around 6 to 8 weeks for the growth to appear, right? And why is that occurring? Because of the slow growing nature of Mycobacteria species, and that too because of longer generation time, that is around 10 to 15 hours. So this is an added advantage of using liquid cultures over the agar-based medias, right? So we get the results within 3 to 4 weeks, and if in case we need to give the report, a negative report, we have to wait until 6 weeks to give any negative report, to provide negative test results. Another important thing with the use of liquid culture media is that firstly they are going to have the broth, which is majorly 7H9 Middlebrook 7H9 most of the culture media uses, and along with it there are certain nutrients. So these are enriched with certain nutrients or certain other growth medias. Along with that, they have certain antibiotic mixtures as well. Antibiotic mixtures, they are not going to allow the growth of unwanted bacteria, unwanted pathogens or unwanted bacteria. So they do not interfere with the results. So this is the reason why we add antibiotic mixtures, and even normal flora doesn't grow. So the detection gets easier. So now let us talk about the automated culture methods. So firstly we have the very famous 𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏. 𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏, followed by 𝗧𝗘𝗟𝗘𝗟𝗏. So these methods are not just detecting the growth of mycobacteria, but some of them also detect resistance of any particular anti-tubercular drug, right? So let us first discuss about the midget 𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏. So this midget stands for Mycobacterial Growth Indicator Tube. And it works on one principle where it uses oxygen sensitive quencher system, fluorescence quencher system. So there is oxygen quencher system which is used in this particular method. So what exactly happens? Suppose this is the culture media, this is the culture tube containing the media, and the broth, it is mixed with certain fluorescent material, some fluorescent dye plus broth. So now this fluorescence is not visible initially. Why? Because this broth contains oxygen fluorescence quencher system. So initially there is a lot of oxygen in the media, right? But when any bacterial growth occurs, if any bacterial growth occurs, this oxygen gets utilized. So now since the oxygen had quenched the fluorescence, it was not allowing the fluorescence to appear. So once the oxygen gets used or it gets utilized, then the fluorescence appears in case of any bacterial growth, and that too mycobacterial growth, right? So how do we detect that? We detect that with the help of observing this particular fluorescence. So are we able to appreciate this fluorescence in second tube, this yellowish area, in the third tube as well, and in this sixth tube? So these tubes are showing growth of mycobacteria, and that is why they fluoresce. So this is the principle on which the midget test or midget method is based upon. Similarly, we have other tests as well that are 𝗏𝗏𝗏𝗏𝗏𝗏 and 𝗧𝗘𝗟𝗏. So 𝗏𝗏𝗏𝗏𝗏, it uses the detection of carbon dioxide which is released, and this is also done with the help of fluorescent material and LED light. It somehow allows the fluorescence to appear, and it is majorly based on the utilization of carbon dioxide. One important thing, midget not just detects the presence of mycobacterial growth, it is also used to detect resistance. resistance if any. And the 𝗲𝗏-TREC, it is based on the measurement of the head space pressure. So it is also dependent on the metabolic activity which is produced, the pressure in the upper portion of the bottle which are connected inside the instrument or inside the machine. So they are going to detect the pressure which is generated because of the metabolic activities of the bacterial growth. So the pressure rises because of the metabolic activity, and this pressure is measured inside the machine, and this is the principle on which 𝗲𝗏-TREC works. So BacT-Alert is just measuring the mycobacterial growth, whereas 𝗲𝗏-TREC, it also detects resistance plus growth. Right? So now let's move on to the molecular methods. Firstly, talking about their advantages. What is a major advantage of molecular method over conventional culture methods, whether it is liquid or any algae based medias? So firstly, they are highly sensitive. Then of course they have very short turnaround time. That means you can get results within a few hours, 2 hours. Some machines have a turnaround time of 1 hour, etc. So you get very quick results, right? So with the help of molecular methods, you are easily able to detect any resistance, right? Then along with the resistance, these are also used for epidemiological purposes as well. What kind of epidemiological purposes? So these are typing of certain new species can be done, right? So these are the major advantages of molecular methods over conventional methods. So these tests are very useful for extrapulmonary samples as well. Now let us move on to the molecular methods which were either earlier in use or they are currently being used. So firstly we have the nested PCR. So nested PCR was the most commonly used and most widely used molecular method which targeted insertion sequence 6110 gene. It is not used now. We have a lot of better advanced options available. Then we have the next important molecular test, that is line probe assay. So line probe assay, it is based on the principle of using probe based detection of amplified DNA in the samples. So it uses certain probes for detecting the amplified DNA. So what are the benefits of using line probe assay? What are the uses? So firstly, this particular test is used to detect the Mycobacterium tuberculosis complex, MTB complex, first thing. then not just detection of MTB complex, but they are also going to speciate Mycobacterium tuberculosis, speciation of MTB, and non-tubercular microbacteria also, right? Along with speciation and identification, they are also going to help us detect the presence of any resistance to first or second line anti-tubercular drugs. So they are going to detect resistance to first as well as second line anti-tubercular drugs. So these are the important advantages associated with line probe assay, or benefits or uses. So one major disadvantage with line probe assay is that firstly it is very expensive, and the second thing is that it can be used only and only for positive culture or smear positive samples. Smear or culture positive samples. So now let us discuss about CBNAAT. It is also known as GeneXpert. CBNAAT, it is the cartridge-based nucleic acid amplification test, and GeneXpert has been endorsed by WHO. Now, this is based on the real-time PCR. Real-time PCR format. And what exactly is it detecting? Firstly, of course, it is going to detect the MTB complex. Along with MTB complex, it is also going to detect rifampicin resistance. Rifampicin resistance. And this resistance is due to mutation of which gene? 𝗙𝗵𝗆 gene. So it is going to use this particular format. It uses 5 probes for 𝗙𝗵𝗆 gene. And the turnaround time for CBNAAT is very, very less. That is within 2 hours, turnaround time. Within 2 hours, you get the results. And along with the detection, it is definitely providing us an added advantage of giving rifampicin resistance also. So CBNAAT principle we have already used. It is based on the principle of real-time PCR. And it is giving us the 𝗙𝗵𝗆𝗏 gene mutation detection. It is using 5 probes for this particular gene and uses, as already discussed, MTB complex and rifampicin resistance. What is the detection limit of CBNAAT? So it can detect as low as 131 bacilli per ml of sample, sputum sample. Turnaround time is very less, that is 2 hours. So we also have an advanced GeneXpert method which is known as 𝗕𝗵𝗆𝗏𝗏, which has the detection limit of as low as 16 bacilli per ml. One thing, and along with this low detection limit, it also uses 2 sequences, insertion sequence 6110 and insertion sequence 1081. So these are the two added sequences that 𝗕𝗪PH uses. So this is the diagrammatic depiction of how CBNAAT or this GeneXpert machine works. So initially, we have to liquefy the sputum and inactivate with the reagent, sample reagent. So it is 2:1 ratio, 2 parts sample and 1 part is the reagent. So we have to mix it, and then we are going to transfer this particular mixture, this particular concoction in this cartridge, into the test cartridge which comes along with the machine itself. So we are going to measure and transfer around 2 ml of sample, and then the cartridge, it is inserted into MTB-RIF test platform. RIF stands for rifampicin resistance. So this is where our work ends. No hands-on after this. This is where we are not supposed to touch anything in the machine. So once this sample is mixed in the reagent and it is loaded in the cartridge, so this cartridge is then inserted or put inside the machine, which flags which portion, which space is opening up, which space is free for the new test to appear. So then inside the machine, sample is automatically filtered and washed. Then lysis of the organism occurs and free DNA is released. Then the DNA molecules are mixed with the dry PCR reagent because it is based on the real-time PCR format. Then amplification and detection in integrated reaction tube occurs. So this particular amplification is being done inside the machine itself, and finally we get the results. So the results can be printed. They are available in software, and it says for this particular test, it says MTB detection low and rifampicin resistance is not detected, right? And the time to result is roughly around 1 hour 45 minutes to 2 hours. So this is how we load and unload and read the results of CBNAAT or GeneXpert. And then we have better advanced version of Truenat, of CBNAAT, which is known as the Truenat. So what is this? Is the particular setup. This is the whole machine of Truenat. So this is now available in certain areas or centers where air conditioning and uninterrupted power supply cannot be maintained. So this is a chip-based format. Chip-based format. Chip-based PCR. So it is battery operated and it is portable as well. Portable. And the turnaround time for this particular machine is just 1 hour. So we have a chip-based format wherein we are going to detect all the resistances present where MTB positive case is there. This is going to detect. This is the cassette where everything, the sample reagents are mixed, then the chip is going to detect. The PCR or the amplification is going to be get done, and finally we'll get the results in the machine itself. So NTEP is now endorsing this Truenat in a lot of areas, a lot of centers. So till now we were discussing about the detection of active Mycobacterium tuberculosis infection. So now let us briefly discuss about the detection of latent TB. And it can be done by 2 methods, that is tuberculin skin test as well as IGRA, interferon gamma release assay. So these latent TB diagnoses can be done based on the delayed hypersensitivity test, which is majorly the principle of tuberculin skin test. It is used for detection of latent TB infection. And what is the antigen used here? It is purified protein derivative. Purified protein derivative. So it contains, we are going to use 1 tubercular unit of PPD, and we are going to inject 0.1 ml of this particular antigen intradermally into the flexure surface of forearm. Flexure surface of forearm. So 0.1 ml of PPD is injected intradermally into the flexure surface of forearm, and the results are read after 48 to 72 hours. And how are we going to read the results? Firstly, we are going to look for the induration surrounded by erythema. So how do we read the results? So since we have already injected the PPD into the skin or intradermally, so we are going to read the result with the help of a scale, measuring scale. And how are we doing it? We will measure it horizontally. If suppose we get induration in our forearm, so we are going to read the results horizontally. We'll put the scale horizontally and we measure it. We are not going to measure it vertically. And what are exactly we measuring? Are we measuring the erythema as well? Are we measuring erythema or induration? So firstly, we must know what induration is. Induration is hardening. It can be very well palpated as well. And with the help of a light, we can very well see the elevation which is present above the skin surface. Induration can be palpated, and erythema would be redness around the induration. So we are measure the induration. Induration, not erythema. Erythema is not being measured, right? So now let us interpret the results. If there is none or an induration less than 5 mm, then this is a negative result. If it is 6 to 9 mm, then we consider it equivocal. And if this induration is more than 10 mm, then we consider it as reactor. TB reactors or positive. Right? So this is how we read and interpret the result. And by the way, what is the name of this particular test, this tuberculin skin test? It is known as Mantoux test. Mantoux skin test, which is a delayed hypersensitivity test. Delayed type or type 4 hypersensitivity. Right? So what are we interpreting and what do we infer out of such results? Firstly, if this test comes out to be positive in case of an adult, then we interpret it as there has been an exposure. There has been an exposure to MTB. That does not necessarily mean that the patient has active TB. Which is exactly the opposite case in children, where such induration is considered as a positive test, positive result in case of children, and this is regarded as a active TB. Then comes false positive results. What can lead to false positive results? This can be due to non-tubercular mycobacteria as well, rarely. Then this can occur post-vaccination as well. And when do we get false negative results? We can get false negative results if it is a miliary TB case, miliary TB. If it is detected too early, the test is done too early or too late. So these are the cases wherein we are going to interpret the result according to the different categories. So now let us discuss about the next test, that is IGRA. So IGRA stands for interferon gamma release assay. So this particular test, it uses two specific antigens. What are those antigens? These are 𝗓𝗏6 and CFP10. So 𝗓𝗏 stands for early secreted antigen, antigenic target, early secreted antigenic target, and CFP stands for culture filtrate protein. So how do you remember these two antigens? This is a very volatile thing to remember. So it is just the mnemonic which says I and E sat in a coffee parlor from 6 to 10 p.m. So I, E sat, CFP 10 and E sat for 6. So this is how you remember it. So what is the procedure that we follow in this particular test? So what exactly are we doing? We are going to collect sensitized T lymphocytes from suspected patients. Now, these T lymphocytes would be exposed to these highly specific targets that are ESAT and CFP10. So what happens now? Exposure to these particular antigen leads to a higher release of interferon gamma, a very high level of release of interferon gamma from T lymphocytes, which shows us that this test has been positive or the patient has tuberculosis, right? Patient has or patient was sensitized to this particular test. So this is detecting the latent TB which is not at present manifesting, right? So this has an advantage that it is very highly specific test, and no false positives can be possible. No false positives. Right? So let us look at the procedure with the help of an image. So firstly, what was done? Blood was collected from the individual. So MTB, this is an MTB infected individual. So what happened? The bacteria was presented to T cells by the antigen presenting cell. So this patient has been sensitized to T lymphocytes, right? So now what happens? T cell stimulation is being done in the next step. So how are we stimulating it? Of course, we are going to use these TB specific antigens to trigger the release of interferon gamma. So suppose this is the sensitized T cells and we are going to expose them to these two antigens, ESAT and CFP. So as a result, interferon gamma is released, which can be detected in the tube as well, right? And then this particular number or this particular amount of interferon gamma can be detected with the help of an ELISA reader, right? So this is how we perform this particular test, and this is how we know that this particular patient is positive for a latent TB infection. So advantages we have already discussed, that this is highly specific, and no false positives, no false positive tests or results. So this was all about the major lab diagnostic features, the certain techniques, whether it is molecular techniques, culture-based identification, liquid medias, and all the important tests which are included in TB diagnosis.